Hi, I'm David Linden,
And I'm Julia Brill.
and we're here from the Johns Hopkins University School of Medicine in Baltimore, Maryland, to present our proposal for the 2018 Tom Slick Research Awards in Consciousness. And our proposal is entitled The Microanatomical Substrates That Preserve Consciousness in a State of Hypothermic Circulatory Arrest. Sometimes some of the biggest challenges that the brain faces in maintaining consciousness are the things that we don't even think about. We go to sleep, we wake up the next day, we're still us. Your consciousness, your individuality, isn't erased by everyday experience. We grow, we change. At a chemical level, the proteins that form the connections between neurons in our brain, where we think our individuality and memory are encoded, turn over every few days or so. And yet, in spite of this turnover, we're still us. Our consciousness is maintained for an entire lifespan. Perhaps the biggest challenge to consciousness comes in those rare situations where the electrical activity of the brain can be almost entirely turned off.
This happens, for example, in hibernating animals, but also in cases of accidental hypothermia. For example, a few years ago, there was a case of a 7-year-old girl in Sweden who was swept into the ocean while playing on the beach in the winter. She was found later floating in the water, lifeless, without a heartbeat. And when she arrived at the hospital, her body temperature was only 55 degrees Fahrenheit. In what seems like a miracle, she was actually revived and went on to eventually recover without any lasting medical problems. Hypothermia is also used medically to protect the brain after injury or stroke, and also during some surgical procedures such as aortic arch repair that require an arrest of circulation. Medical hypothermia minimizes the damage to the brain that normally occurs when the brain is deprived of oxygen. So there are really two remarkable aspects to cooling the brain down. For one, cold temperatures protect the brain from damage due to the lack of oxygen. This is most likely because at cold temperatures the brain needs less energy, and so it can withstand periods of low oxygen much better. And secondly, as the brain cools down, all electrical activity in the brain slows down and eventually stops altogether. But then the brain can basically reboot itself afterwards and resume functioning almost as if nothing has happened. So whatever makes us us can be preserved in a brain that's devoid of measurable neuronal activity. We're interested in what exactly happens in the brain when it is subject to cold temperatures and what that tells us about the brain's ability to recover from that. And there are three main things that might happen. First of all, there may be no measurable changes in the brain at all. Secondly, there may be changes, but the brain comes back to normal eventually. And thirdly, there may be a few lasting changes, but the brain has enough built-in redundancy so that these changes can be tolerated.
So ideally, we would love to be able to peer into the brains of humans while they are experiencing deep chilling, and to do it in a way that didn't damage their brains or their skulls, and to do it in a way that would allow us to see individual synaptic connections in the brain and how they change. There's many problems with this, however. First of all, ethically, we don't want to just chill humans down as if they were drowning in the ocean just to satisfy our curiosity. And second of all, the kinds of brain imaging techniques that are available for humans, like fMRI machines, have a resolution of about a millimeter in diameter. Unfortunately, synapses in the brain are about 1,000-fold smaller, about a micron in diameter. So we're stuck. What are we going to do? So we're going to make use of our mammalian cousin, the mouse, which shares most of our brain physiology with us. We're going to use special mice that have been genetically engineered to express glowing proteins in some of the neurons in our brain. And we are going to perform surgery on these mice to cut a tiny hole in the skull and implant a glass window that allows us to look inside. Now, there's still another problem. If you look through this window, the brain isn't transparent. It's opaque. It looks like a boiled egg from the surface. So in order to see down into the tissue and to be able to make time-lapse movies of those synapses before, during, and after cooling, we need a special microscope called a two-photon confocal microscope.
When you look through a cranial window onto the mouse's brain, you can see the blood vessels at its surface. They don't change over time and provide a kind of roadmap to the brain below. Using them as a guide, we can zoom into the same small part of the brain again and again to image the green fluorescent neurons. Here are examples of the same neuronal segments imaged 1 week apart. The structures we study are dendrites, long thin processes that are studded with so-called spines. Spines are the site where synapses are formed and signals are transmitted between neurons. These images, taken over the course of 6 weeks, show how this particular dendrite segment changes over time. Most spines remain and are stable, but a few spines appear and disappear. We're interested to see if these dynamics change after hypothermia. We give our lab mice experimental hypothermia. Here are 2 of our mice before the experiment, and they're exploring a novel cage. Shortly after this, their body temperatures cool down to 15 degrees Celsius, which is about 60 degrees Fahrenheit. While cold, the mice stop moving, their heart rate and breathing slow down, and they barely react to touch. At this point, most of the electrical activity in their brain has ceased. However, only a few hours after warming up again, the mice are active. They don't show any obvious adverse effects from the hypothermia.
The experiments that Julia just described are remarkable in that they allow us to make time-lapse movies of the very substrate of consciousness, those synaptic connections that encode our individuality, our memories that change throughout our life and make us us. In the case of cooling, they allow us to answer the question, does this fine structure change during cooling? Do these synapses fall apart but then reform again upon rewarming? This is crucial to understand the continuity of consciousness in this major disruption. It is our hope that the award from the Tom Slick Foundation will allow us to collect enough preliminary data to ultimately write a grant to the National Institutes of Health to support a more complete study.